We think of our bodies as these amazingly complex machines, right? But we usually think about them in a very classical kind of way.
Yeah.
But um, what if there's like this whole hidden layer,
Mm-hmm.
like some kind of subtle quantum influence that's
Yeah.
kind of always at play, like right inside ourselves?
Right.
And imagine if like even just weak magnetic fields, the kind that are just like all around us anyway, could actually be like nudging our cellular processes
Oh,
in ways that we're only just beginning to understand.
yeah.
It kind of sounds like something out of science fiction.
As a bit, yeah.
But today we're diving into research that suggests this might actually be happening.
Mm.
Like quantum phenomena could be subtly shaping our biology like in our everyday lives. Pretty wild stuff.
Yeah, it really is kind of a mind-bending shift in perspective, isn't it?
For sure.
Like, what I find really fascinating is this growing idea that the macroscopic world of biology, the stuff we can see and interact with,
Right.
might not be totally separate from the rules that govern the quantum realm. It's like those two worlds are kind of blending together.
Yeah.
And you're right, the idea that something as common as a weak magnetic field could have these real effects at the cellular level,
Right.
it really does make you rethink how biological systems work.
Absolutely. All right, so let's dive into the specifics. The research we're looking at today is titled Spin Biochemistry, Modulates Reactive Oxygen Species Production by Radiofrequency Magnetic Fields.
Okay.
It's kind of a mouthful.
Yeah, it is a bit.
But basically, these researchers were looking at how radiofrequency magnetic fields, and these are similar to the kind emitted by our phones, though obviously much weaker,
Right.
how these fields impact the production of certain molecules in rat lung cells.
Okay, interesting.
Specifically, we're going to be focusing on their findings about superoxide and hydrogen peroxide,
Right.
which are super important in how our cells
Yeah.
function.
Absolutely.
Our goal today is to really understand what this study found and what it might mean for the interplay between electromagnetic fields and our cells.
It's like trying to figure out if the electromagnetic environment we live in is actually having a bigger impact than we realize.
Exactly.
Yeah.
Now, you mentioned this quantum biology angle. This isn't exactly mainstream biology yet, is it?
Yeah.
What's the main idea behind it, and how does this research fit into that?
You're absolutely right. It's a pretty new field. Basically, quantum biology explores whether quantum mechanics, those rules that govern how tiny particles behave,
Right.
whether those principles actually play a significant role in biological processes. For a long time, people thought that these delicate quantum effects would just get washed out. Because the environment inside a living cell is so warm and messy and dynamic.
Right.
But more and more evidence suggests that that might not always be the case.
Wow. So there could be these little pockets of quantum weirdness happening inside of us all the time.
Potentially, yeah.
Yeah.
And this study tries to bridge the gap between that microscopic quantum world and the macroscopic cellular world by using magnetic fields to influence magnetically sensitive free radical pairs. Those are molecules that have unpaired electrons.
Right.
And then they observe what happens at the cellular level.
Free radicals. We hear about those all the time when we're talking about aging and antioxidants and stuff. So how do magnetic fields interact with those?
Well, that's where the spin-correlated radical pair mechanism, or SCRPM, comes in.
SCRPM.
Catchy, right. It's the most likely explanation for how weak magnetic fields could influence reactions involving these free radicals.
Okay. I'm listening.
So picture these radical pairs, and they have their electrons spinning kind of like tiny little tops.
Okay, I can picture that.
But these spins aren't random. They're correlated. Their behavior is linked. And weak magnetic fields can actually influence the alignment and evolution of these spins because of quantum mechanics.
Okay.
This little nudge at the quantum level can then affect how long the radicals stay paired, how likely they are to react with each other,
Right.
and what kind of products they form.
Wow, that's pretty amazing. The study mentions that this SCRPM is well understood in photosynthesis,
Right.
which is obviously like the basis of all life on Earth.
Exactly.
And they suggest it might be involved in other things too, like how birds navigate using the Earth's magnetic field.
That's right.
But they also mentioned that there's still debate about this, and that not all research in this area has been consistently reproduced.
Right.
So where does this study fit into all of that?
Yeah, that's an important point. And, to their credit, the researchers are very upfront about the challenges in this field. While the SCRPM makes sense theoretically, and there's good evidence for it in some systems, it's really hard to definitively prove that it's happening in living cells.
Yeah, I can imagine. It's complex.
Exactly. It's really complex. This study adds another piece to the puzzle, though.
Okay, good.
They focused on a very specific process, the production of those reactive oxygen species,
Right.
and showed a correlation with RF magnetic fields.
Okay.
It's not definitive proof, but it's definitely intriguing and warrants more research.
So they were looking at rat lung cells, specifically RPA-SMCs. What did their experiment actually look like? How did they expose these cells to magnetic fields in a controlled way?
So they used what's called an in vitro system, which means they did the experiments in petri dishes outside of a living organism.
Right.
They had a control group of cells that was exposed to a static magnetic field, an SMF,
Okay.
of 45 microtesla. To put that in perspective, that's about the same strength as the Earth's magnetic field. Then they had an experimental group, which was exposed to that same static field, but with an added component,
Okay.
a weak radiofrequency, or RF field.
Hmm.
This RF field was oscillating at 7 megahertz with an amplitude of 10 microtesla RMS, and it was perpendicular to the static field.
Got it. So the key difference was that added RF field in the experimental group.
Exactly.
And what were they measuring in these two groups of cells?
Primarily, they were focused on the production of reactive oxygen species, those ROS molecules we talked about earlier. Specifically, superoxide and hydrogen peroxide. They used some really sophisticated techniques to accurately measure the concentrations of these molecules in both groups.
All right, so let's get to the good stuff. What did they find? What happened to the levels of superoxide and hydrogen peroxide when they introduced that RF field?
This is where it gets really interesting. They found that the RF field caused a decrease in superoxide concentration in the rat lung cells. But at the same time, they saw an increase in the concentration of hydrogen peroxide in those same cells.
Oh, wow. So it wasn't just a simple increase or decrease in oxidants overall.
Right.
It was a specific shift from one to the other.
Exactly.
That's pretty wild. So how did they explain this? What's the mechanism that connects the RF field to that targeted shift in ROS?
Their proposal is that it's happening through singlet-triplet modulation of spin-correlated radical pairs,
Okay.
specifically involving semiquinone flavin enzymes, F80HN, and superoxide.
Right.
Remember that SCRPM we talked about?
Yeah, vaguely.
Well, they think the RF field is interacting with the quantum spin states of those radicals. The idea is that the 7 megahertz RF field causes something called decoupling of flavin hyperfine interactions.
Flavin hyperfine decoupling. Okay, now you're just using big words to scare me. Break that down for me.
Huh, all right, fair enough. So flavin is this molecule that's crucial in tons of biological processes.
Right.
And hyperfine interactions are basically about the tiny magnetic fields created by the nuclei of atoms
Okay.
and how they interact with the spin of nearby electrons.
Right.
So the researchers think that the RF field is somehow messing with this very delicate dance of magnetic forces at the quantum level,
Okay.
specifically between the flavin and the superoxide.
And how does messing with that lead to less superoxide and more hydrogen peroxide?
It all comes down to the concept of singlet and triplet spin states in that radical pair.
Oh, I remember those words from earlier.
Right. So immediately after an electron transfer happens, this radical pair is thought to be mostly in a triplet state. Now, the triplet state makes it more likely that those two radicals will just diffuse away from each other,
Right.
and you end up with superoxide as a separate molecule.
Gotcha.
But if the RF field helps the pair transition from that triplet state to a singlet state, then the radicals are more likely to react with each other while they're still close together.
Okay.
And this singlet state reaction pathway leads to the formation of hydrogen peroxide.
So the RF field is kind of like a quantum traffic cop.
Exactly. It's subtly influencing the direction of the reaction. More singlet state means more hydrogen peroxide and less superoxide.
Very cool. Now, all of this relies on some pretty precise measurements, right?
Yes.
How did they actually detect these changes in superoxide and hydrogen peroxide to support this idea?
They used a combo of really sensitive techniques. For superoxide, they mainly used electron paramagnetic resonance spectroscopy.
EPR.
Right, EPR, along with a special molecule called a spin probe that reacts with superoxide. This creates a product that they can detect and measure with the EPR. Interestingly, they also said that another common spin trap wasn't as effective in their system.
Okay.
For hydrogen peroxide, they used a fluorescence-based assay using a molecule called Amplex Ultra Red.
Got it.
This molecule lights up or fluoresces when it reacts with hydrogen peroxide,
Right.
allowing them to measure its concentration.
Clever. So they saw these changes, but they also did control experiments to make sure these effects were really due to the RF field rate.
Oh, absolutely. Control experiments are crucial.
Right.
Like when they added catalase, an enzyme that breaks down hydrogen peroxide,
Yeah.
they found that the RF-induced increase in hydrogen peroxide was suppressed.
Okay, so that shows that increase was actually because of more hydrogen peroxide being produced.
Exactly.
What other controls did they do?
Well, they used something called PEGS-ODE, which is a type of superoxide dismutase,
Hmm.
an enzyme that converts superoxide into hydrogen peroxide.
Hmm.
And they saw a decrease in hydrogen peroxide in the RF-exposed group.
Okay, that makes sense if the RF is already shifting things away from superoxide.
Exactly. It all fits together. They also use DDC, which inhibits superoxide dismutase.
Right.
And as expected, they saw an increase in hydrogen peroxide.
Okay, so that further supports the link between superoxide and hydrogen peroxide.
Right. They even looked at the effects of paraquat and DPI,
Okay.
which can influence ROS production in other ways, and they found that the RF fields seem to be acting independently of those.
Interesting.
And maybe most importantly, they found that the RF field alone reduced the baseline superoxide concentration by a whopping 40%,
Wow.
compared to the cells that were only exposed to the static magnetic field.
That's a big difference.
It is. And when they used paraquat to further suppress superoxide,
Yeah.
the RF field actually boosted that suppression. A similar thing happened with DPI. It increased superoxide,
Right.
but the RF field brought it back down compared to the DPI alone.
So no matter what they did, the RF field seemed to push the cells towards less superoxide and more hydrogen peroxide.
That seems to be the trend, yeah.
Pretty convincing. Now, the study didn't stop at just measuring ROS, did it? They also looked at cell proliferation, how fast the cells were growing and dividing.
You're right, they did. And this is another really fascinating finding.
Okay, what did they see there?
They found that the RF magnetic fields actually made the rat lung cells proliferate more.
Really?
Yeah, they saw an increase of up to 45% compared to the control group.
Wow. So the RF fields were somehow making the cells grow faster?
It seems that way. And importantly, they didn't see any significant changes in cell viability.
Meaning the cells weren't dying off more quickly.
Exactly. They were just growing and dividing more readily.
And they think this increased proliferation is related to those higher hydrogen peroxide levels.
That's their hypothesis.
Okay, how does that work?
Well, it's known that low to moderate levels of oxidative stress, often caused by molecules like hydrogen peroxide,
Right.
can sometimes trigger signaling pathways that actually promote cell growth.
That's counterintuitive. I always thought oxidative stress was a bad thing.
It can be at high levels, but at lower levels it can act as a signal.
Interesting.
They think the RF-induced shift towards more hydrogen peroxide might be stimulating these growth pathways in the cells,
I see.
leading to the increased proliferation.
This brings us back to those flavoenzymes we were discussing before. The researchers think these enzymes are really important for this whole spin biochemistry thing.
Yeah,
Can you explain a bit more about why?
sure. So flavoenzymes are a huge family of enzymes, and they're involved in tons of reactions in the cell,
Right.
especially those involving oxidation and reduction.
Okay.
Many of them use flavin cofactors like FAD.
Okay.
The study specifically mentions a reaction scheme proposed by Vincent Massey, which describes how reduced flavoenzymes can interact with oxygen to produce superoxide as an intermediate.
Okay.
The researchers believe that it's at this stage,
Yeah.
when you have that radical pair between the reduced flavin and the superoxide, that the RF field is having its effect.
I see.
By messing with the balance between singlet and triplet states in that pair,
Right.
the RF field can then change the outcome of the reaction and what products are formed.
Fascinating.
And they even suggest that this might not be limited to just the NOx enzymes.
Which are the ones that produce superoxide.
Right. They think this could be a more general thing happening with other oxidases, too.
Wow. So this could be much more widespread than we initially thought.
Potentially, yeah.
Now, obviously, this study opens up a lot of interesting questions.
Absolutely.
What did the researchers themselves point out as areas that need more investigation?
They were very clear that their study mostly provides indirect evidence for this spin biochemistry mechanism. They really emphasized that we need more research to directly look at the spin states of these radical pairs in real time inside cells.
Right. Like actually see what's happening at the quantum level.
Exactly. They suggested doing more experiments where they change the parameters of the RF field, like its orientation, strength, and frequency,
Got it.
to see how those changes affect ROS and cell proliferation.
That would really help confirm their findings.
Absolutely. They also said we need to investigate other sources of ROS in the cell, like the mitochondria,
Hmm.
and use different inhibitors to target specific pathways more precisely.
Right, because there can be other things going on that we haven't considered yet.
Exactly. And one really important limitation they mentioned was that they didn't do a true off-resonance experiment.
Not bad.
Basically, it means using an RF frequency that's deliberately not at the right resonance for those hyperfine interactions to see if the effects disappear.
Okay.
That would provide much stronger evidence that it's really a specific quantum interaction that's causing these effects.
So this study is a great starting point, but it needs more work to fully nail down what's going on.
I think that's a fair assessment.
Given that everyone listening is constantly surrounded by RF fields, what are the broader implications of this research? Why should someone who's not a scientist care about this?
Well, it raises some big questions about the non-thermal effects of RF exposure.
Right, because usually we only worry about things like tissue heating.
Exactly. But this research and other work in quantum biology suggests there might be other more subtle mechanisms happening at the molecular level.
And those could be important because we're exposed to RF fields constantly.
Exactly. From a public health perspective, we need to understand these effects better.
For sure.
Plus, if we can really figure out how EMFs interact with biological systems, it could lead to some really cool new technologies in medicine and bioelectronics.
Like using EMFs to control cellular processes?
Potentially, yeah.
That would be amazing. All right, so to wrap things up, what would you say is the main takeaway for our listeners?
I'd say the key takeaway is that this research shows pretty convincingly that even weak RF fields can affect important cellular processes. In this case, the production of reactive oxygen species. The proposed mechanism involves this subtle manipulation of quantum spin states,
Right.
which leads to changes in superoxide and hydrogen peroxide and even cell proliferation.
Yeah, for me, the most mind-blowing thing is that these quantum interactions, which seem so abstract, could be having real measurable effects in our bodies,
Right.
just because of the magnetic fields that are all around us.
It's a pretty profound shift in how we think about biology.
Absolutely. And even though this study focused on just one cell type and one RF frequency,
Right.
it suggests that we're only just beginning to scratch the surface of understanding these interactions.
I agree there's so much more to explore.
So to everyone listening, as you go about your day surrounded by all these invisible EMFs, think about this. What other processes in your body might be subtly influenced by these fields in ways that we don't yet fully understand? And what might the long-term consequences of that be?
That's a great question to ponder.
It really makes you appreciate the connection between the quantum world and our everyday lives.
Absolutely. It's a connection that's becoming more and more apparent all the time.